kinds of cells. So this Fc gamma R1A is expressed on all these cell types, and so on. Almost all of them have many different cells expressing these Fc receptors. So you think you give a patient an IgG, and where is it going to go? First of all, it will find its target, but then what's it going to do? Which of these cells is it going to, which receptor is it going to find? And if it's going to find a receptor, which cell type is that receptor going to be in? And actually, very little is known about this. And, well, we certainly think that this inadvertent binding to different cells, it could well explain some of the serious side effects that you get when using these monoclonal antibodies. So I'm predicting that in the future, you will not only have to test the activity you want, you're going to have to show that your monoclonal antibody does not bind to receptors on cell types which give you inadvertent responses, which could be very dangerous side effects. So ADCC itself, it's now understood that if these sugars here do not contain core fucose, you actually get more effective ADCC. And this is because if you take core fucose out of here, this junction it's clear, it's less cluttered, and there's movement of one or two amino acids here, which enable this Fc gamma R3 receptor to find the top of the CH2 domain on the IgG. So this is IgG. These are we tip this sideways so you could see the binding better. These are the FAB regions. This is the FC. Here are the two conserved sugars. These are the ASM 297 sites. And here is the Fc receptor binding portion, which is more tightly binding here if you free this junction from the core fucose. So you can see here, if you have no fucose and you have a glycosylated receptor, you get much more binding and activity than if you have no glycosylation of the receptor and you have core fucose in the way. So all of this amounts to your need to be able to regulate glycosylation both in the process and also to be able to monitor it in the product. So what we tried to do in our lab is to design a glycoanalytical platform that was fully comprehensive. So it could take you all the way from very detailed analysis of glycoproteins to understanding their function to doing basic research, all the way to doing cell line selection, to checking that your highly producing cells actually have appropriate glycosylation, to checking very simply for whether you have antigenic epitopes there, for following your production, and in particular scale-up, because we know scale-up sometimes changes glycosylation, and all these different things which affect the media. We also needed to be able to have process analytical technologies to do at-line monitoring, because sometimes if you can see the glycosylations going wrong, you can do something about it. You also need to have very effective glycosylation analysis for these critical features, and also for testing function, as I've just shown you. So, product stability is also important. So you can see there are many different reasons why you need to do glycoanalysis, and you don't use the same methodology for every case. So what the sort of analysis you might do here will be very different from the sort of analysis you'll do if you're just looking for alpha-gal residues, or if you're just tracking a well-known process analytical technology. So the basic platform that we have is like plug and play. You can use the whole thing for very complex analysis, or you can take pieces of it if you want to do something more simple. Now, I just showed you some of the factors in production which control glycosylation. But of course, that isn't the only thing. There are a lot of different things that control glycosylation. So the genetic profile of the cell line is really important. The proteome, so are your proteins effectively expressed, and do they have the right sugars attached to them? And this all involves many pathways within the cell. And the quantitative traits like the yield, the robustness, the type of glycosylation, these are all the same kind of things that you would ascertain if you were looking at natural glycoproteins from natural cells. And then you will have a complex cell phenotype, which you also will have naturally, but you will have it in your cell culture cells as well. And each of these steps are accompanied by a lot of things that can alter them. There are 600 proteins that are involved in glycosylation. So if you change the genetic profile of many of these proteins, you will affect glycosylation. Epigenetics is important. If you start to demethylate, you will change, for example, sialic acid content. Cytokines, lipids, metabolites, metal ions, change the levels of those and you will affect glycosylation. Cell death, as we just saw, autophagy, hypoxia, post-cell proteins, upscaling, all of these affect glycosylation, pH, temperature, dissolved oxygen. So not only do these things affect the 3D protein structure, they also affect the glycosylation. So this, when you get into what's happening in a bioprocessor, is a very complex set of processes. And we can get some sort of handle on what's going on from the work that we do on natural proteins. So this is human serum IgG. So we took, I think we've taken 10,000 people now, and we've removed the glycans from their polyclonal IgG, and we've run a glycosylation profile for each of those people. And then we've integrated under the peaks, and we know from our analysis what's in each of these peaks. Then we've taken that integrated value and we've linked it to SNPs in the genome of each of those people. So we found that particular levels of particular glycans link to SNPs in an individual's genome. And the significance of these is very high. So by doing this, we uncovered a lot of genes that all associate with different IgG glycosylation. So, for example, the top 5 hits are these genes here, and we know that they affect silation, they affect this Icarus factor, affects glucosylation, as does FUT8. Genes here which affect the presence of the dolichol phosphate precursor, the building of this precursor, the attachment of the bisecting glucnac, and the attachment of galactose. So some of these are genes that we had no idea affected glycosylation. So there are many things which we really still don't understand about control of IgG glycosylation, from the genome, the transcriptome, the metabolome onwards. So now, in order to avoid all this problem of media control, or make it simpler, people are really now using glycoengineering. And this is some work that's done by Sui Song and Pei Qing in Singapore. So they have used different techniques to make homogeneous glycosylation. So this is just to make sure that there's no fucose on the sugars so that you can promote ADCC. So here they've made a lectin-based selection, but more powerfully really now, we can use gene editing tools like zinc finger, like Talon, and now CRISPR. All of these are very effective ways of actually deleting the enzymes that are required for glycosylation or the transporters in order to get homogeneous glycosylation for a particular therapeutic function. So we've made a series of cell lines here, each of which have homogeneous glycosylation processing. We can make them fucose-free, sialic acid-free. We can have man-terminated glycans. We can make highly silated EPO. We get less heterogeneity, and we get advantages in bioprocessing. And if you do the glycosylation analysis of these, cell lines, you can see here that there's the fucose has been removed by using this glycoengineered version. I'm just going to. So another way of gene editing is to just make single point mutations. If you make a point mutation at this alanine residue, this residue here, then you can affect methylation. So an individual, just changing an individual amino acid can sometimes be very powerful in changing methylation. And I'll just go. So one of the problems, as I said, in using non-human cells is that you can lead to get serious side effects, and this is just to show you how serious it can be. This is the first cetuximab that came out. It was produced in a mouse cell line. It had FAB glycosylation, and what people didn't realize was it had this alpha-gal structure on it, and this is the result. Really serious side effects from this. And if you start to use sugars which have N-glycosyl-neurominic acid instead of N-acetyl-neurominic acid, again you risk having these serious side effects in some people. So I hope what I've shown you is that we need to be able to understand how to analyze glycans at all these different stages of bioprocessing. And tomorrow I'm going to do a workshop, so I'm going to go in more detail into the glycosylation analysis. So I'm just showing you just two trailers. This is our automated glycan sample robot, which we use to prepare the glycans. We then put fluorescent labels on them. We run them through an LC and a mass spec online, and then we have programs which automatically integrate the data, compare them with the database, and allow us to get glucose units values, which we saw just now. We use the enzyme digests, and then all of that is confirmed by the mass spectrometry that we do online. So this technology now, it's a good time to do glycan analysis because this technology is really it's really come of age, and it allows you to do all kinds of experiments in a very straightforward manner. So looking at the way the glycosylation is sensitive to the bioprocess, so looking at different levels of silated glycans, you can now do all this with a programmed robot. You can monitor the amount of these nanostructures which signal that the bioprocessing is not going very well. And many other factors are now really possible with these new technologies. So I acknowledge my group in NIBIT in the beginning. This is my group in Singapore who also produced some other data. So thank you very much.
Since there is a seminar scheduled for noon, I'm going to take 2 questions. And also we, of course, have tomorrow for further discussion. Jackie?
Thank you very much. We know that we could obtain different glycosylation patterns in a protein at the same time. But do you think that glycosylation could be controlled by variation in tertiary structure of the protein, or maybe certain events that affect the time residence of the protein in Golgi?
Yeah. Yes. Yes, absolutely. The structure of the protein. That's why you have site-specific glycosylation, because the structure of the protein affects the glycosylation differently at each site. And the residence time is also really important. If you do it at low temperatures, for example, everything goes through more slowly and it changes the glycosylation, it changes the site occupancy as well sometimes. So the Golgi enzymes, as I'm sure you know, they're very carefully arranged because everything has to be sequential. So if you see the sialic acid residue, sialyl transferase before you see the galactosyl transferase, you won't be able to silate because the galactose won't be there. So the organisation of the Golgi and the length of time that a protein has to stay there to make sure it interacts with each of the enzymes is really important.
Yes. What happened with the antibody chimeras? Some of them have enhanced cytotoxicity activity. Mm-hmm. It's complicated to generate this, or?
Yes, well, they have to be designed to do what you want them to do. So if you want them to be cytotoxic, then you have to, engineer them so that you promote cytotoxicity with their FC region. And if they're designed with an FC region then it's an anti-TNF receptor on the top, for example, you want something to happen with the FC. But sometimes you don't want anything to happen with the FC. So people even use non-glycosylated FC sometimes, just because they don't want any function, but they just want the FC region to be there to present the active group. So I think it's just all of these different.
Thank you very much, Pauline. Thank you.